studies involving the, studying of inheritance, and they involve the collection of the germline DNA, which is the DNA taken. So you take a blood sample from the patient and then derive a DNA from it, assuming that all of the mutations that you'll find in this kind of DNA would be the mutations that a person got from its parents. So, but what appears to be is we sequence this cell. So DNA in blood comes from this myeloid cell line, and what could happen? So originally, I don't want you to get through the whole of the scheme. The left-hand side of the scheme is what kind of I want you to understand. So that originally we have a multipotent stem cells. So those are the long-term hematopoietic stem cells and the short-term hematopoietic stem cells. So long-term hematopoietic stem cells, those are the very original cells which give this give rise to all of the possible blood cells that we have, and the events of they're being renewed pretty rarely, so that could be once in several months. But what they constantly are producing are the short-term, so-called short-term hematopoietic stem cells, which then are differentiated in all possible kinds of the blood cells. So this is the step. This is kind of the key step where so-called the clonal expansion events could happen. So here is an example. So how such events that are mosaic and what are those could arise. So this is an example of the progression of the disorder called the myelodysplastic syndrome. So this is a disorder affecting the bone marrow and thus a production of the long-term hematopoietic stem cells. So in a normal case, so what you have is the normal differentiation of the long-term hematopoietic stem cells. So they produce these multipotent progenitors and then the final kind of differentiated cells. So in the case of low-risk myelodysplastic syndrome, so this orange bar is just a piece of DNA that harbors a mutation predisposing to the myelodysplastic syndrome. So what happens is there are a lot of the progenitors produced. So instead of differentiating of the single cell, but the idea is that then you still have exactly the same number of copies of each of the certain cell. So they all will have. So if you will take the sequence, the case A, which is normal, you'll see a normal count of heterozygous mutations. So you'll have half of the reads telling you that there is a heterozygous, that there is an alternative read, half of the reads telling you that there is a reference genotype. So in case of B, you'll see the mutated DNA, you'll see a mutant with still a normal count of the alternative and reference alleles. But now, in case of the high risk of the myelodysplastic syndromes, there is an abnormal number of the long-term hematopoietic stem cells which have this mutation predisposing to myelodysplastic syndrome. They're actually replicating much faster than the normal wild-type long-term hematopoietic stem cells. And because of this, they could abnormally differentiate, producing a certain number of clones, so-called, harboring the predisposing mutation. So if you would take case C and sequence that, then you will see that there is way more reads supplying the information that there is an alternative allele standing in inherited DNA, which would result in a so-called perturbations in the allele balance. So what allele balance is just the fraction of the alternative reads for a given genotype, right? So every time you sequence a certain base pair, so you will see that there is a certain number of alternative, certain number of reference reads. For heterozygous, the expectation is 1/2, right? So you would expect that half of them would be alternative. If you see only 10% say of alternative reads, then now you can ask a question whether this is such an event of the clonal expansion, as we can see on the on the part C of this slide, or what the technical challenge is that this could be just a mistake of the sequencing, right? So that would be just a, some kind of artifact. So this is where the technical challenge comes from. So such mutations which have an abnormal allele balance resulting from, assuming they're resulting from the clonal expansion, we will call them mosaic events, right? So none of us inherit such things from our parents. So this is all what happens during the human's life. So the original motivation for this study comes from the original study of such a clonal expansion events and clonal hematopoiesis in leukemia and other blood cancers performed by Giulio Genovese, who is also a co-author on the paper that I'm presenting, and he's actually working here with us at the Stanley Center. So what this paper in New England Journal of Medicine tells us is that such mutations, such a mosaic events, are actually very strongly correlated with the with the progression of the leukemia, so development and progression of the leukemias and lymphomas, and moreover, it has been shown that they're actually bringing a causal role for that, which is a reasonable thing. So this is an event taking place in blood, so somehow affecting the bone marrow. So that intuitively could be, quite clearly linked to the blood cancers. What then means, so the 4 genes have been identified out of this paper. Those are PPM1D, TET2, DNMT3A, and ASXL1. Interestingly, that I think all of them, so it's not clear, completely for DNMT3A, but all of them have a certain role in the tumor suppression. So PPM1D and the ASXL1 are the important members of the respectively BRCA1 and BAP1, which are known cancer genes in the complex and interactions with those proteins. So then a reasonable question would be to ask if such, the same effect could be observed for the solid tumor cancers, which is not as trivial because technically those mutations would arise only in blood, right? And you don't know per se anything about the tumor. So it has been recently shown by Elise Ruark and her co-authors that mosaic events in PPM1D are actually associated with the higher risk of the breast and ovarian cancer. Moreover, it has been shown that in this paper, so that it's specifically the very last exon of PPM1D affected by mosaic protein truncating variants would be actually leading to the higher risk of the cancer development. What's interesting is that after performing some experimental validation, they actually observed that the changes in the last exon of PPM1D sequence actually lead to a higher rate of affection by the ionization radiation on mice. So kind of trying to suggest the pathway which would be, linking the causal role of such mutations in PPM1D. Though nothing has been shown about TET2, DNMT3A, and ASXL1 in that perspective. So basically, our question was, can we, try to study those kind of mutations in these genes in people who have only solid tumor cancer and don't have any evidence of the blood, any kind of leukemia, lymphoma, or other kind of situation. So the design of the study originally included about 6,000 controls. Those are coming mostly from ESP studies and diabetes studies. So those are people without any known cancer phenotype. So at the time, they are fairly old. With the I'm going to show the age and mean age and other data about the sample. So we're quite confident, say, about the fact that they either don't have cancer at all throughout their life, or they have a very low risk of cancer because they're old and they did not develop it at the point of sequencing. So, and as a cases, so we needed a large cohort of the cancer samples, which is the most famous example of that is the Cancer Genome Atlas. So we had almost all of those samples available, so it's roughly about 8,000, and we collected about 20 solid tumor cancer phenotypes, so all kind of tumors. So, and the most important part is that for each of the samples, it's the germline, so the blood DNA sample is available, and also the tumor DNA sample is available. So we know both mutations from the tumor and from the blood. So the important part, as I mentioned in the beginning, is that at some point, because of such a low, low count of the alternative reads supporting the genotype, mosaic events quite often could look like they are actually, a mistakes of sequencing or sequencing errors. So you should be, very careful about discriminating between the two, between the two scenarios. So, and moreover, since coverage becomes very important to discover such mosaic events between cases and controls, and what we want to do is to compare the burden of such mutations in cases and controls, it's very important to confirm the uniform distribution of the coverage. And what actually appears that they're very similar. So what you see on the plot is the probability to observe the protein truncating variant depending on the coverage in both cancer cases and controls. And we actually, how we define the the mosaic events in such cases. So we looked for an unusual distribution of the alternative reads supporting the heterozygous genotype with the binomial test, and setting a hard threshold on the coverage. So it must be at least 20x. And even besides that, so we observed that controls are slightly better covered than cases, and that's why we actually even thus adjusted for the any coverage differences as a also we use the coverage as a covariate in our case control analysis. So sorry, I forgot to mention this, but that might be one of the questions that would arise. So both the cases and controls were so how we discovered the genotypes in them. So they were all sequenced here at the broad, and then brought to the same exact alignment pipeline, and they were jointly called. So we're confident that there are no batch effects or discrepancies between the genotype coding between the cases and controls. So the other thing you want to control for is the age of the samples. The reason for that is the following: that mosaic mutations, with no even respect to cancer or any other phenotype, so the clonal expansion events were are shown to be highly associated to the age. So the older, usually you would expect those kind of events to arise, roughly about after 50 years old. So the reason for that is kind of, on the background, the reason is intuitive, that since the long-term hematopoietic stem cells are renewed very, quite occasionally. So what happens is you need, multiple multiple replication cycles until the moment when, just a random mistake arises in the long-term hematopoietic stem cells DNA. So that's usually the reason for the late onset of such mutations. And thus we controlled for the age of cancer and the control samples. Well, there is a certain discrepancy. The controls are, fairly old also, but on average, the cancer cases would be slightly older. So we needed to adjust our null hypothesis about the distribution of the mosaic events between the cases and controls. And that's how this plot would look like. So that's the, what you see on the plot is the probability of finding a mosaic protein truncating variant in cancer cases versus controls. And what you can see is that the probability after the age of roughly 60, the probability of finding a mosaic protein truncating variant in cancer sample is much, much higher than in controls. So we basically adjusted the null distribution, so the expectation of the burden of the mosaic PTVs for the for this kind of age dependence. So all of the further studies are adjusted for age, and so the other thing is, so you see two p-values on the plot. So one of them corresponds to all 20, all 20 phenotypes, cancer phenotypes that we had. The other one is actually mentioned that it's without the breast cancer and ovarian samples. The reason for that is, as I shown you earlier, so in the RARC paper, they already shown the signal of association of breast cancer and ovarian samples to the mosaic mutations in PPM1D. So we wanted to test whether that is the reason for for this association, or there is something else additional to that. And clearly, you can state that there is an additional signal to the PPM1D mutations. So then, a reasonable question would be to ask, what's the reason for such mutations to arise? And the idea is the following. So first, you want to ask a question whether they are the early markers of the developing cancer. So those are basically the mutations that are either causal to the tumor onset, or they might be very early, since those are white blood cells, right? So that might be an early response of the immune system to the a very, very small tumor progenitor arising in the organism. And what we did, we screened about 800 early onset cancer cases. So all of the people in this cohort had the onset of cancer less than 35 years old. And the statement is that there is no mosaic mutations burden compared to the same controls. And the trick is that it's actually very few mosaic events at all that you can find there. So it's very atypical for people to have the the mosaic mutations such early in life. So clearly that rejects the hypothesis about the any time in life causal event. So it's definitely not causal in terms of the early onset cancers. So and then the other hypothesis is that to test that for old enough onset, so like in the cancer in 60s or 70s, like in TCGA. So that's another pitfall. To clearly answer this question, you would need the cohort of samples that would be sampled. So you'll take the DNA sample before the cancer onset and after the cancer onset, and then see whether the burden of mosaic PTVs is different or not. Well, it's a very tricky thing to ask for. So it's a very, there's no such cohort. What we tried to do to answer this question, I'm gonna cover this a bit in a few slides, I think. So what we did, we tried to look on the large set of the unascertained for cancer population. So like about 12,000 of samples from the Swedish biobank. It actually appears that it's very hard to make this. So the frequency of the mosaic PTVs is such a low that if you take the unascertained for cancer population, it's very hard to make a conclusive statement even having 12,000 of samples. But I'm going to talk about this a bit later. So, and another interesting observation is the following. So what you see on the plot is the allele balance for heterozygous protein truncating variants, which we found to be mosaic. And once again, just to remind you, the allele balance is the frequency of the alternative alleles. Sorry, not the frequency, the fraction of the alternative alleles supporting the heterozygous genotype. So it's basically the number of the alternative reads divided by the total number of reads covering this particular position in the genome. So what you see on each plot is that that each bar is representing a single sample. So if you look on the ppm1D plot, which is the top left, so this bar is the single individual having a bladder cancer, and gray bar is basically the fraction of the alternative reads in a blood DNA, and the blue bar is the fraction of the alternative reads in a tumor DNA. So what you can see mostly is that the frequency of such a mosaic event, so the fraction of such mosaic events observed also in tumor is actually equivalent to just a blood contamination of the tumor sample, right? So you'll always have a certain, small amount of the blood contaminating the tumor sample due to the way you obtain that sample. So for some of the samples, you can actually even confidently say that there is no such event in a tumor at all. And given the fact that the tumor DNA is usually sequenced much more deeply than the germline DNA, that's even more surprising. So usually the tumors are sequenced at a coverage more than 100x. So it's actually quite a robust and surprising finding, which once again asks an interesting question: if those mutations are actually linked, as we observe, they're linked and somehow associated to the solid tumor cancers, they're actually not present in the tumor. So it's actually quite an interesting observation. I'm struggling to give you a reasonable explanation for that, unfortunately. So, you can see consistently observe this for all of the genes that has been shown to be linked to the cancer progression. The other hypothesis is whether this could be induced by the cancer treatment. So since, as we discussed in the beginning, the long-term and short-term hematopoietic stem cells are being produced in the bone marrow, and you know that majority of the methods of treatment of cancer actually involve the radiotherapy, which definitely affects the bone marrow functionality, right? So here, I'd say our test is very inconclusive because of the following. So the data present in the TCGA and available through the TCGA portal is very noisy with respect to how exactly those people were treated. So there is first a lot of the missing data, and second, as I figured out, there is a lot of discrepancies in that data. So despite the p-values are inconclusive, we cannot tell whether this is in any way linked to the to the chemotherapy or radiotherapy. There are several papers, I think they were published early this year and in the end of the last year, suggesting that this is the clonal expansion and mosaic events and the solid tumor cancers are actually driven by the chemotherapy exposure. So there is a paper that tests the amount, so basically how what's the burden of the mosaic events based on the number of cycles of chemotherapy that the patient received. And there is a clear correlation in that. So that would be also, explanatory in some way to the higher risks of the leukemia and lymphoma in these patients. So, but so far, as in I can only speculate on the statement, and I cannot make I just don't have, and I doubt anyone else has enough data to definitely make a conclusion about this. So the other interesting part is you would expect under the null hypothesis, right? So if you expect that the mosaic events just happen and they don't have any biological relevance, or this is just a result of the DNA damage due to chemotherapy or radiotherapy, and that does not bring any functional biology in here, then this statement could be questioned with this slide. So what is here is that on the left you see the mean age of each of the cohorts. So these are the codes for TCGA. So this is a head and neck squamous cell carcinoma, breast cancer, bladder cancer, and so on. So those are the codes for the phenotype. So all those are different kinds of phenotypes. And you actually see that there is a clear association of the genes to a specific type of the cancer phenotype. So what we did not reach the the genome-wide significance, or the because we lack some cases. So the story that I've shown you with the Ruark paper about the association of the breast ovarian cancer and PPM1D mosaic protein truncating variants. So we actually see that in ovarian cancer, we would have a burden only of PPM1D mutations, but not the other ones. So it's a very specific kind of gene to cancer phenotype association. The same you would see in glioblastoma and lung cancer. And so basically there is a certain burden of the gene to a cancer phenotype correspondence. So that suggests a certain type of the hopefully biological reason for discrimination and functionality of those genes and linkage to a certain cancer phenotype. So as I mentioned a bit earlier, so we tried to replicate our study and actually try to answer the the causality question in the following sense. So we had about 12,000 of samples from Swedish national patient registers, and what those are. So those are the people of different age, but they have a pretty well documented all the phenotypic information about them. So we can figure out how many of them had cancer, what kind of cancer, when in life, what kind of treatment they received, and so on. So the only difference is that out of those 12,000, so those are 12,000 of people unascertained for any kind of disorder. So the frequency of the cancer in these 12,000 of samples would be very similar to what you can estimate from general population. So only about 5 to 10% of this cohort would have any kind of cancer record. And what's even more interesting, so they were followed up after the DNA sampling. So with such a cohort, we were hoping to answer the question, if we see the mosaic PTV, mosaic protein truncating variant in one of those genes, can we actually then get back to that patient and see whether in 3 to 8 years the patient actually developed solid tumor cancer? And that kind of answer we cannot get because apparently either the follow-up time is just not sufficient enough, or another thing, we just don't have enough cancer cases in this cohort due to low incidence of cancer. So our test is not conclusive. But if you look on the following data, so out of those 12,000 samples, we took all of those samples who had a prior DNA collection record. So we excluded all leukemia and lymphoma patients. We actually observed with the same panel stage, as you see, the higher is the older the person is, the higher is the burden of diabetes. But you actually can see that the incidence of cancer, prior background of cancer, all the years, like is actually higher than in all cases. So it has been shown also that the TPA is more affected by mosaic incidence mutations, so that's true in the general medicine, showing a certain type of the TPA incidence mutations that mosaic affecting the clonal expansion events rather than the protein truncating variants, and we should observe that this still apart. According to the leukemia and lymphoma, and the NMT3A gene harbors most of the mosaic events in this gene. Same actually is true, so if you had a chance to look at the preprint of the paper, so there is actually figures that each of those genes is actually very specifically affected by the mosaic mutations. So after the all of the region of the third of the so all of the mosaic plants are sharing enough with certain factors usually. So basically, what we find from here is as a summary. So first, to answer the question of causality or chemotherapy, or basically to answer the question, okay, so to see the association of why, we actually need to set the post-DNA collection follow-up, which was basically to always answer the question, what happened earlier. So basically, all the truncated mutations are for the cancer phenotype and cancer. So the other thing is that the clearly observed association of the same genes that are responsible for leukemia with the solid cancer phenotypes, and the interesting novel mutations actually found in the tumor. So there is a big interest in cancer gene specificity, which once again, so far has been the observation, and we definitely know that the concept of cancer for cancer population, there is a significant burden of the 3 DNA collections with areas. So with that, I would like to end the story to acknowledge my goals main to work. So I don't know if you take any questions if you might have them.
Yeah, you can stop sharing the screen.